Very good morning, students. We are going to study in class 11th chapter Cell Theory and Cell as Unit of Life. Children, according to the latest course, reduced course, we are going to study only certain topics. We are going to study only certain topics of this chapter in detail, but not all the topics. Like students, cell theory has been deleted, but I will not teach cell theory in detail, but then we have to study eukaryotic cell in detail. Again, prokaryotic cell is reduced in the syllabus. So let us start with this chapter, student. Name of the chapter is, student, Cell Theory and Cell as Unit of Life. Very simple definition. You are studying from your basic class. Cell is a basic structural and functional unit of life, isn't it? Now students, cell theory has been deleted in the reduced course. So let us check once. It was formulated or postulated by Swan and Schleiden, but extended or contributed by Rudolf Virchow. And the postulates are as follows. Cell is the basic structural unit of life. Cell is the basic functional unit of life. Simply we can divide the definition: basic structural and functional unit of life. Simply we can divide it into two parts. And the third is, student, all cells arise from pre-existing cell. Omnis cellula a cellula. So this is all about, students, cell theory. Now the next topic in this chapter, the cell theory and cell as a unit of life, is eukaryotic cell. Student, you know very well what is the difference between eukaryotic and prokaryotic cells. Eukaryotic cell, student, the main difference is because it consists of true nucleus. Children, true nucleus means nuclear material is bounded by nuclear membrane, isn't it? This is eukaryotic cell. Okay, and these are the important organelle. This is, we have drawn eukaryotic animal cell. So we have centrioles or centrosome, isn't it? And these are these extending bodies from the nuclear membrane up till cell membrane is your endoplasmic or cytoplasmic reticulum. And children, these red dots, granular bodies, are nothing but ribosomes. So endoplasmic reticulum with ribosomes on their surface is called RER, rough endoplasmic reticulum. And in some cells, when the endoplasmic reticulum are not covered by the ribosomes, granules of ribosomes, then those are called SER, smooth endoplasmic reticulum. And student, these are the other organelles. This endoplasmic reticulum, they get pinched off to form Golgi apparatus. This Golgi apparatus in the plant eukaryotic cell is called dictyosomes. And this is your powerhouse of the cell, mitochondria, cytoplasm, and this is, student, your suicide bag, lysosomes, isn't it? So student, why this cell is eukaryotic cell? Because in this it consists of true nucleus, in which the nuclear material is bounded by nuclear membrane, isn't it? So this is all about your eukaryotic cell, student. Okay. So student, according to the reduced course, structure of prokaryotic cell is not there, but there are some important structural features of prokaryotic cell, student, which will help you to compare and study eukaryotic cell. So just let us see few features only. Like prokaryotic cell It is the same figure studied by you in Monera, isn't it? In classification of biological classification. And then student, this is the main feature which we are going to study. We compare eukaryotic cell with this part. This is not true nucleus. It is simply naked or unbounded nuclear material, isn't it, student? And that is called nucleoid. Nucleoid, what is important for your NEET exam. And then second important feature of this cell important for your NEET exam is plasmid. Student, plasmid is extra chromosomal circular DNA, and this is also absent in eukaryotic cell. And the third one which is absent in eukaryotic cells are sex pili. These are the bodies, extended bodies, sex pili, which helps in the reproduction. That is also not in the eukaryotic cell, isn't it? And then mesosome, student. Mesosome, this cell membrane invaginates. This green color cell membrane, it invaginates like this, and this is called mesosome, and this helps in the division of the cell, asexual reproduction, isn't it? So this is just a glimpse for prokaryotic cell, not in detail because it is reduced in the course. Okay children, as I told you that it's necessary to compare the eukaryotic cell with prokaryotic cell, isn't it? These are the features of eukaryotic cell. Children, the first one, as I told you, it consists of true nucleus in which nuclear material bounded by nuclear membrane, isn't it? Here just opposite, here lacks true nucleus. Then DNA is linear, spiral and associated with histone octamer. Student, you know histone protein. Histone protein or histone octamer along with DNA in eukaryotic cell forms a complex called nucleosome. Okay, so these are the characteristic feature of eukaryotic cell. Then another characteristic feature of eukaryotic cell, it can organize to form tissue, but here no organization or formation of the tissue. And last but not least, children, here all important cell components or organelles are present in eukaryotic cell, isn't it? And the best example, EU, eukaryotic, euglena, algae, protists, fungi, plants and animals, except bacteria, isn't it? So these are the four features of eukaryotic cell. Now children, we are on very interesting topic of the chapter cell biology. That is the portion of the syllabus is your cell envelope. Children, this is cell envelope, isn't it? That is called cell membrane or plasma membrane, common in both plant and animal cell. So student, this cell membrane name was given by Sir Kramer and Nageli, but this fluid mosaic model was proposed by Singer and Nicholson. Okay student, this fluid mosaic model, this one was proposed by Sir Singer and Nicholson. So let us study in detail children. You see this student, this matchstick like layer, two layers, this one upper and just opposite and parallel here the lower one. This is called children phospholipid bilayer. It is made up of phosphate and lipid molecule, phospholipid bilayer. And children, when we see one unit in detail, this one here like this I have drawn, this is the polar head. Polar head, it is hydrophilic in nature. Okay? It is hydrophilic in nature, and this tail is hydrophobic in nature. Okay? Here after PHILIC will be there. Hydrophilic. Okay? Hydrophilic means children water loving. Okay? Water loving. And hydrophobic tail means student water relucting, isn't it? So this is all about your phospholipid bilayer. Okay, now the next part in this student, this green color, are you observing this green color? Student, these green colors are nothing but globular proteins. This protein, this green color which is present superficially attached to the outer surface of the lipid bilayer. These superficially, these are called extrinsic or peripheral protein, isn't it? And this protein which are partially or completely embedded in the lipid bilayer is called, children, intrinsic or integral protein. These are the globular protein, student. And what are their nature? These extrinsic proteins, student, they are soluble and can readily dissociate in the water or any other solvent. Okay? From the membrane they get readily dissociated. And children, these intrinsic protein are not soluble and cannot easily dissociated. Okay? From the membrane. Okay? And the best example of this extrinsic protein, student, extrinsic protein is your cytochrome oxidase and ATPase. These are the enzyme and they are protein. And the example of intrinsic protein, student, is integral or intrinsic protein, student, are spectrin on the surface of RBC. Okay, student. And now the third and the last thing which we are going to study in this important part of this fluid mosaic model is, children, you see this dotted chain structure. This, are you seeing this? So student, this is glycoprotein chain. Okay? So glyco and protein means, student, protein with attached carbohydrates. Protein with attached carbohydrates, isn't it? Glyco means carbohydrate and protein with attached carbohydrate. This chain consists of. And student, this is glycolipid chain. That is lipid with attached carbohydrate. Glyco is common. So carbohydrate is common. And here protein with the attached carbohydrate, and here lipid with attached carbohydrate. And student, these between these two integral protein, there is a pore formed by integral protein. So student, this is all about your fluid mosaic model proposed by Sir Singer and Nicholson. And these are the two differences what I have dictated you orally about the location and nature. Location and nature. And this is one unit of this bilipid layer, phosphobilipid layer, polar head and tail. Okay, student.